Hi everyone, my name is Jasleen Bhatia and I am a junior currently majoring in public health in the Blaustein School of Planning and Public Policy. So this summer I was given the opportunity to conduct research in Project Super with the West Lab, which is a part of the psychology department here at Rutgers. This summer my work was primarily consisting of counting microglia cells of various brain regions in order to understand the results of our ongoing project. Our work focuses mainly on opioid addiction, which is a major issue that impacts millions of individuals across the globe. And this disorder is correlated with many major diseases and compromised immune responses. So from previous work and literature review, we know that human brain imaging during fentanyl administration reveals that the effects of fentanyl use are highly localized to areas of the brain which are involved in reward and addiction. And in Figure 1, we are looking at the total drug overdose deaths in the U.S. in 2016. And as you can see, over 40% of them are related to any opioid, but 24% are related to prescription opioids. So this is just showing the prevalence of this problem and why our research is very applicable to today. So previous work has also shown that non-contingent injections of fentanyl use in rats shows an immunosuppressive effect within 24 hours of drug abstinence, and these subjects are more susceptible to infections and illness even though their physical effects of their drug use have left. So this experiment is unique in the sense that it uses a different type of model. We're using the self-administration model, which is the closest to human drug-related behavior. And then this experiment is seeking to understand the severe immunosuppression effects and if this lasts after a specific period of time. So in order to understand the effects that opioids have on the immune system, we need to look at cytokines, which help mediate the type of immune response we have to several different things, one being drug use. So we mainly focused on IL-1 beta and TNF-α, which are secreted through microglia cells. And in our experiment, we had two groups, one receiving saline and the other receiving fentanyl. And this experimental phase lasts for 30 days where they are just receiving infusions. And then they could either receive a month of abstinence or a week of abstinence. And then both groups are treated with lipopolysaccharide. So to properly identify the microglia cells after the subjects are anesthetized, we use a phenotypic marker called anti-IBA1. And so we took 16 slices from each animal from a variety of regions in the brain. A few to mention would be VTA and NAC. And these slices were imaged and uploaded to Fiji, which is a software that allows us to properly visualize the cells. So each of our cells were counted by two members of our lab. And in Figure 3 here, we are looking at the NAC region. And as you can see, there are several microglia cells, and we are specifically interested in counting the cell bodies. Because of COVID-19, our lab had to close for some time, so we were unable to perform other analysis of our results. But by completing this analysis, we're hopeful that we will be able to better understand how to treat individuals that are going through withdrawal and abstinence periods to minimize relapse. And this research has implications for better understanding the role of opioid withdrawal and the effects on the immune system. So I would like to thank the Project Super for allowing me to be involved in this research, and my PI, Dr. Mark West, and my grad student, Angela Dow, for helping me over the past few years in learning more about addiction. Thank you.